Hello, everybody. In this lecture, we are going to talk about the replication of animal viruses, which is in contrast to the previous lecture on bacteriophages. So let's get right into it. By the end of this module, you should be able to describe the generalized steps for animal virus replication, compare and contrast how enveloped and naked animal viruses differ in their replication pathways. Alrighty. And so when we think about um, virus replication in animals, it's very similar to virus replication in bacteriophages. You've got attachment and penetration. However, there is a step called uncoating, which is new, followed by replication, assembly, and release. And so for attachment, this can occur a couple of different ways, whether it is a naked virus or a enveloped virus. But generally, regardless of what type of virus, the host cell is going to have a receptor that is going to attach to a protein on the virus. Now, that may be a capsomere if it's a naked virus, or it could be a spike on one of your envelope viruses. Regardless, something is going to attach to that host receptor cell. And then you're going to have penetration, where the viruses are going to um, fuse with the membrane if they are enveloped, or be endocytosed if they are naked, and sometimes if they are enveloped. But regardless, one of these two things are going to happen. You're either going to have membrane fusion, which can only happen if you have a membrane, or you're going to have endocytosis, which can happen whether you are already membrane bound or not. Then you have an uncoating step. And so in the uncoating step, your enzyme is going to be digested by um, enzymes that are in your endocytic vesicles. Or potentially it can happen in the cytoplasm, or it can happen in the nucleus. In the image shown here, you see that the virus that penetrates the cell is going to be carried in an endosome, which is ultimately going to combine with a lysosome, which is going to facilitate the uncoating process. Now, this is just like before, you're going to have some sort of replication. This is where the genome is going to be replicated and viral proteins are going to be made. But keep in mind that this happens using the host's cellular machinery. Um, Very few viruses package any enzymes in their capsid, um, and all viruses rely on host machinery to some extent to allow replication to occur. And then lastly, you have assembly, and this is where your genome is going to be packaged into your um, capsids, and you can be released back out of the cell. So this can happen through budding, where your envelope viruses they travel to the cell membrane. They are going to bud out where the envelope surrounds the capsid, and then this is how they become enveloped. On the other hand, you could have basically a lytic cycle where the viruses are going to lyse the cell and release all the viruses at once. All right, so that should be very similar to the bacteriophage lecture. Um, you should be able to describe these general steps of virus replication in animal viruses and compare and contrast how this differs between the enveloped and naked viruses. With that, I hope you enjoyed, and I'll see you soon.